in our webinar this morning, the importance of good data management practices in clinical trials. My name's Nigel Kirby, and I'm the Head of Data Management in the Centre for Trials Research. So this morning we have the following agenda. So we'll be talking about what data management is. We'll look at some definitions of data management, why it's important and how it fits into a clinical trial. We then have Helen talking to us about how we get good data for clinical research. Mia is then going to talk to us about the regulatory frameworks for data management and how these frameworks bind our work together. Ceri is then going to look at CTR databases and validations. So look at the databases we use and how we validate these. Debbie's then going to talk about CTR examples of good practices, how we are ensuring that our data management methods are constantly evolving. And then finally, Kim is going to talk to us about using routine data in trials and how we're more frequently using these methods. And then hopefully at the end, we'll have some time for some questions and answers. So firstly, what is data management? So whenever I present, I always like to use this definition. So it's the process by which the required data is acquired, validated, stored, protected, and processed, and by which its accessibility, reliability, and timeliness is ensured. So there's a little bit of a myth around data management that it starts when you have your first piece of data to manage. But actually, that's not true. So data management starts long before a study or a trial opens, as you need so much goes into the planning of the trial. So planning how you're going to handle your data, what data you're going to capture, how you're going to capture it, how you're going to store it. So actually, as part of resourcing data managers, we actually put vast majority of our resource at the start of the study, so we make sure that we get all of our processes correct. So why does data management matter? So it's all about having confidence in your data. So everything that we do in CTR and all of our outputs rely on good data to be able to look at if we're going to be changing policies, changing the ways that people implement our practices. So we need to be confident in our data to be able to implement these. So data management, there's lots of different strands to this. As I mentioned, planning is key. So writing things such as a data management plan. So this details everything that is needed, how our data is collected, monitored and checked. Looking at ethics, legal policy, looking at things such as our general data protection regulations, GCP, the HTA if we're using samples, and also making sure that we can justify all of the data that we're collecting. We don't want to be asking participants in our studies to be giving us lots of different data if we're not actually going to be using this. So how are we going to be capturing our data? Is this going to be through databases? Will we be using paper CRFs? Will we be using wearable devices? So we need to be considering all of that. So then we also need to be thinking about where's our data going to be stored. We need to make sure that it's secure at all times. You know, we have to comply with, as I say, GDPR to make sure that participants' interests are at the forefront of everything we do. We're also having a lot more cases now of reusing data. So researchers may come to us in the CTR to say, we've seen you've done this study, we've collected this data. We actually think that we'd be able to answer our research question by using your data. So we need to make sure that all of our data is in a usable format. So this is the data lifecycle of a clinical study. So we start off with the protocol design, and in that protocol, it will say what our research questions is, what our primary and our secondary outcome are, and what data we're going to be collecting to be able to achieve that. So then we move through to our case report form design and what we call in the Centre for Trials Research our metadata design. And some of the presentations later on will touch a bit more on this. But this is essentially building up our questions that we're going to ask in the study. We then move to using this metadata and our CRFs to build our databases so that when the researchers, either in the field, in hospitals, or participants are answering these questions, these will likely be on our database. So our database development team take this, they build the database, and our data managers then make sure that they validate this database before it is used by researchers or the public. This validation is still what we call user acceptance testing. So making sure that everything that we've put in our metadata design and our CRFs is contained within the database. We'll then move on to our data collection. So this again will be by participants, by sites, but they'll be using that database. We've got the data entry still on there. This is mainly if we're using paper case report forms, and they would come back to the Centre for Trials Research to be entered. Our data managers then will work on data cleaning. So this is making sure that any data that's maybe missing, maybe is out of ranges that we would expect, or maybe doesn't fit with the logic of the forms, we would go back and we would query that to make sure that we've got the most up-to-date and accurate data possible. We would then lock our databases. So by doing that, that just means that we remove access so that no further changes can be made after our statisticians and our data managers are happy with the data. And then we move to our statistical analysis and our report writing. So I'm going to stop sharing my slides now, and I'm going to hand to Helen, who's going to take the next section.
Thanks, Nigel. Bear with me. Can you all see those slides? Just nod if you can. Yeah. Yes. Oh, thank you. Yeah. Okay. Yeah. So good morning, everybody. I'm Helen. I'm also based in the Centre for Trials Research. I'm a data manager on an adolescent wellbeing trial, and I also oversee the data management of my colleagues working on research studies within the population health division. So my first slide here, I'm going to go right back to basics and talk about why we as data managers in the Centre for Trials Research are so invested in clinical research data management. So implementing care practices within our current health systems should be supported by evidence. This evidence is derived from the practical implementation of the outcomes of clinical research, but this evidence all started out as data. So between the planning and collection of research data and the analysis, so leading to evidence in which can change the way that healthcare is managed, that's all down to data management and the handling of that data. And so as a result of this, we want to ensure that that data is as good as it can be, so essentially has a high level of data integrity. So data integrity is a concept and a process that ensures the accuracy, completeness, consistency, and validity of data. Data collection with clinical research can be extensive. Good data that is data with high integrity, relies on effective and robust data management processes. And when we talk about data management in clinical research, a lot of what we're talking about is the planning of data collection, the data collection itself, the organisation and processing of this data, and then the storing of data safely so that it can be used for data analysis. And as data managers in the Centre for Trials Research, we often focus on the collection and processing of research data. Sorry, I hadn't switched to the next slide. So as an example here of collecting research data would be recording a person's medical history when registering them onto a clinical trial, and then using a case report form. And then an example of data processing in this context could be numerous. It could mean swapping the participant's name with a unique identification number to maintain their anonymity in the study. It could mean transferring the information from the paper medical history form to a database for secure storage, or it could mean adding and saving the medical history data in the database, which could trigger a planned message on the database stating that the participant is now eligible to be randomised into the study. And this just demonstrates that a good deal of data processing could have happened before a participant has even been randomised into a clinical trial. So Mia will focus on these frameworks for best practice in her slides, but I've just added on here an example of some of the ethical and regulatory guidelines that we use as a starting point to guide data management best practice. So we also use our own standard operating procedures, the study protocol, and a data management plan. And we also need our colleagues and team to support us and guide the way we collect and process research data. So I'm going to show you now what some basic data management strategies to collect and maintain good data looks like, and what practical function they provide. And I've put these under 4 subheadings. So firstly, participant protection. So safety, dignity, confidentiality. And the first example here is to show you how we process participant identity. So it's the participant's right to ensure that the information they choose to share with us is kept safe and confidential. And we translate this in data management as the process of de-identification. And it's pretty straightforward in principle. So we plan our data collection. So we capture name or any other identifiable information such as address, phone number, email address. We capture this once, no more if we can help it. And that just means that the less frequently or the fewer times we capture this information where there's an opportunity to identify the person, the lower the risk of that participant being identified. So you can see on this screenshot here, we've captured the parent's name, the parent in our imaginary study, and they're called ABC123. Couldn't think of anything more imaginative, I'm afraid. So we've captured that in the registration form, but from any data form after, as you can see at the bottom of the screen, they're known as participant 2. So participant confidentiality is essential for good data management. So nextly, part of getting good data is ensuring we get the right data we need to answer the study question, and that will feed into the evidence base for future medical practice. So rather than data processing, this part of data management is about the planning of data collection. And I've put some screenshots on here of our database where we have planned the data collection to reduce the risk of ineligible people being involved in the study. So just to summarise, effective planning of data collection can really help in ensuring that the correct data is collected. And then on to the next slide. So again, similarly, getting good data is about planning to identify and prevent human error. So if we collect the wrong type of data, it can render that data, that potential piece of information completely meaningless. So if we can anticipate the possibility of error, we can prevent it, which means we gather better quality data. And then for this kind of my 4th slide about the functions, I mean, these are by no means exhaustive. There's so many functions to data management, but One of these is also study storytelling. So that's tracking the who, what, when, and why of the participants through the study. And getting good data means some of the processing around is around enabling us to communicate the story of our data, our participants' data as they journey through the study. And this allows us to see if we're getting enough of the data we need to answer our study questions. And so my example here is the collection of 3-month questionnaire data from participants. And we've processed this as data managers by transcribing which participants have completed their 3-month questionnaire. And we do that by using the date questionnaire completed as a proxy for whether there is any data present. And so this is a quick and effective way to quantify completeness, which can then be plotted on a chart, as you can see at the bottom of the screen, which allows us to see at a glance whether the 3-month data collection is going as expected. Are we getting a decent percentage of returns? And so processing it in this way allows us to visualize whether completion rate is good or poor. If it's poor, we can intervene and step up efforts to collect the data. And so they're just summarising, getting sufficient complete data is also an integral part of getting good data. And just to summarise here, good data has high integrity and it's accurate and as complete as it can be. And we maximise the chances of this by employing robust data management practices, including planning data collection and effective data processing. And I've just ended here with some quotes from my colleagues about what good data management looks to them and what helps them on the way of ensuring a good data set. So thank you for listening, and I will now hand over to Mia.
Thanks, Helen. So just bear with me while I share my screen. Can everyone see that?
Yes.
Brilliant. So hi everyone, I'm Mia. I'm also a senior data manager at the Centre for Trials Research, covering the brain health and mental wellbeing division. So I'm going to talk a little bit about some of the regulatory frameworks that are required for high quality data management, and some of the processes we follow at the CTR to ensure our work's compliant with these. So a crucial element of good data management is adhering to the relevant research governance and data management regulations. Some of these are a legal requirement, and this can differ depending on the type of research you're carrying out. So for example, if you're conducting a clinical trial of investigational medicinal product or CTIM, where you're testing a new medicine or medicinal treatment, they're more heavily and legally regulated than non-CTIMs, trials where you aren't. So these frameworks are in place to ensure that the safety and rights of participants are protected, to maintain data integrity, and to ensure that research is being managed legally and compliantly. So we need to ensure that these regulations have been considered when we're managing research data. So what are the frameworks that we need to follow within data management? So General Data Protection Regulation, or GDPR, is one, which I think most people will have heard of. So GDPR is a legal framework, and the fundamental purpose is to ensure that individuals' personal information and data is protected and processed lawfully and safely. So personal data is just any information that can identify an individual either directly or indirectly. GDPR applies to all research studies that use personal data from individuals, and it needs to be followed by law. And then it specifies that all personal data must be processed lawfully, fairly, and transparently, be kept to the original purpose, be minimised, have the accuracy upheld, be removed if it's not necessary, and be kept confidential and the integrity maintained. So within data management, we're frequently managing participants' personal data. So we need to make sure each of these aspects are considered when planning and carrying out our data management processes. Next is good clinical practice, or GCP, which is a set of internationally recognised ethical and scientific quality standards for conducting clinical research that involves people. So these principles focus on ensuring that the rights, safety and well-being of research participants are at the forefront of research. Research is scientifically sound and described in a clear protocol. Quality systems are in place for managing the data, and that information is managed in a way that allows its accurate reporting. So up-to-date GCP training and compliance is a legal requirement for all individuals who work on a CTIM, but it is recommended as best practice for all staff involved in any research trials. So next is the Medicines and Healthcare Products Regulatory Agency, so MHRA, which is a UK government agency responsible for regulating clinical trials that use medicines and medicinal devices. So this applies to CTIMPs only. So they're required to carry out inspections within organisations conducting CTIMPs to assess compliance with clinical trial regulations. Ongoing approval is needed from them to conduct research within CTIMPs. So they may stop a clinical trial from continuing its processes if they aren't compliant. And then within data management, MHRA regulations relate to GCP adherence, management of participant safety data, and ensuring that the necessary data management processes are in place so that data is complete, accurate, and reliable. And then lastly is the Human Tissue Authority. So the HTA independently regulates organisations that use human tissues for research. So for example, at the CTR, many of our cancer studies collect blood samples as part of the trial. So the HTA will inspect organisations and provide guidelines to ensure that human tissue is being used within research trials safely and ethically. So data management processes here mainly involve around ensuring we have full documented consent and continued consent from the research participants for the use of the samples they provide for the study. So those are some of the frameworks we need to follow to ensure high quality data management, and also that we're conducting research legally and ethically. But what does this look like in practice? So this is by no means an exhaustive list, but just to give some examples. Firstly, we need to have adequate procedures to keep the participant informed. So correctly obtaining and documenting informed consent from participants, as well as any requests from participants wanting to withdraw from the research, including in relation to any samples that they've provided. We also need to consider what data is being collected and if it's necessary. So each clinical trial will have a study protocol document, which is effectively the blueprint for conducting the study. So we need to ensure we're meeting all of the protocol data collection requirements and not collecting any data that isn't necessary. So at the CTR, we carry out a data justification process by documenting the justification for collecting each item of data in our studies, just to check its necessity for the study and stop us collecting too much data. And then another important consideration is how data will be kept confidential and secure. So as Helen mentioned earlier, one example of how we can do this is by using participant IDs instead of participants' personal information to keep their data confidential, and limit the personal data we collect to what's absolutely essential. So both electronic and physical data should be stored securely, limiting who has access to authorised personnel only. And then if we're going to share the data with third parties, just ensuring that data sharing agreements are in place, that it's being shared compliantly. And then lastly, data needs to be kept accurate and attributable. So again, access management is a crucial element here. So we'll only give access for authorised staff to the trial database and data. And their access permissions should be in line with the agreed role they have for the study. Having a clear and validated audit trail is also really important to know who's provided what data and when, and if any changes have been made to the data and why. And we can improve the accuracy of data as well by carrying out quality control checks and data cleaning. So this can be writing automatic checks into the database to check for potential errors when the data gets entered. and also manual checks and generating reports for missing or potentially incorrect data. So at the CTR, we have a number of processes to ensure that these procedures are being followed and that we're compliant with data regulations. So firstly, we have standard operating procedures for data management. So these documents just set out in detail each of the processes and steps we need to follow when completing our data management tasks. And they're regularly reviewed and go through an approval process to make sure the information is up to date and in line with current regulatory requirements and best practice. For every study, we also write a data management plan. So that sets out all of the data management processes that need to be followed for each particular trial. And they also get reviewed regularly and require sign-off. So yeah, as I've just mentioned, we have study documentation reviews. So most of our data management documentation undergoes review and sign-off, which is carried out by independent data managers as well as other teams within the CTR with specialist knowledge, such as information system specialists, trial managers, statisticians, and our quality assurance team. So it just means that information is thoroughly checked and everything described is following the right process. And then lastly, as I just mentioned, we have a dedicated quality assurance team. So their job is particularly important in terms of making sure the work we're carrying out is adhering to the required regulations. So they mainly carry out reviews of our documents and processes, advise on regulatory issues, and conduct internal audits of our data management processes and studies to ensure quality and compliance. So finally, just in summary, the regulatory frameworks and bodies are in place to ensure that all research data and reported results of research are credible and accurate, maintaining the integrity of data, and that the rights, safety, and confidentiality of participants in clinical research are always protected. So we need to ensure that the relevant regulations and frameworks are being considered when planning and conducting research, particularly for CTIMPs or studies collecting human tissue samples where more legally required regulations apply. And then lastly, high quality data management processes should be implemented and regularly reviewed to uphold these regulations, ultimately protecting both participants and the data. So thanks very much for listening. I'll now hand over to Ceri for her presentation.
Thanks, Mia. Good morning, all. So hopefully I've started sharing my slides. Can everybody see them?
Yes, thank you.
Yeah, lovely. Okay, so my name is Frayne. I'm the Senior Data Manager for the CTR Cancer Division, and my section will focus on the use of databases and validations. So the aim of any data management process is to provide a high quality and appropriately clean final database suitable for statistical analysis. Extracting some keywords from GCP, there needs to be accurate recording, handling and storage with systems that must ensure the quality of the trial. So this presentation will give a very brief overview on how the CTR designs and builds databases, examples of databases we use and their application in 2 case studies. So metadata is most commonly described as data about data. It forms the data dictionary, which is used to design data collection tools and support the build. Metadata is an external and independent record of all values and validations, which is used for review and testing. It ensures data minimization. So each piece of data must be justified either because it's stated in the protocol, so this could be primary or secondary outcome data, so for instance, eligibility, regulatory data, so safety data, or is required for management of the trial. CTR metadata also includes a system specification. So examples of details include the method of data collection, so whether it be electronic or on paper, identifiable data fields. So this is sensitive personal data which needs restricted access and checks to ensure it doesn't export. Randomization requirements, user and form access, site and participant identification, default options for reasons for data entry changes, and missing codes, and the data lock functionality required. So the CTR data manager is usually the author of the metadata, but we work closely with database developers, trial manager, statistician, chief investigators, and PPI. An internal peer review of metadata is required before seeking final sign-off. So as part of the approvals process, particular emphasis is placed on the variable label, data type, and value label, ensuring PPI review, especially for patient-facing forms such as questionnaires. The internal peer review ensures that the metadata conforms to design best practice and implements the protocol and any mitigations on study risk assessment, such as cross-checking eligibility criteria, endpoint data, and restricting access to sensitive data to comply with GDPR. It also ensures that data exports are clean and complete by ensuring missing codes are provided and validations ensure adherence to protocol. So, for instance, with eligibility checks, as you can see at the bottom of the slide. So CTR has developed a metadata library where similar data capture can be adapted. This slide shows an example of a DEF form from the library and how the template is completed. So questions, or the variable label, value labels, so the category options, aid data entry and can be exported. Primarily for data export for analysis, the variable name, which is the code for the question, and value, so the answer or number assigned to a category option, are used. Missing codes, collect diffs, and validations are built in to promote clean and complete data and maintain data integrity. In this example, an informed validation fires a warning for future data entered. Any form triggers or guidance text can also be added as instructions at the top of the metadata, as per the blue rows, such as to send an email notification to CTR if date of death is completed. These instructions aren't exported from the database. So randomization is the process by which recruited units are allocated to intervention groups with a known, usually equal chance of being assigned to any of the groups. A trial randomization strategy consists of the sequence method, where participants recruited will be allocated to a group, concealment, where the treatment allocated to the next participant is concealed. This ensures any treatment or intervention difference found is genuine, which maintains scientific integrity. Eligibility or stratification criteria for group allocation will be detailed in the metadata. So the statistician will ensure the metadata implements the randomization strategy. Once the trial is recruiting, the randomization allocation data is analysed periodically to check the ratio, and the data is also used in consort diagrams. So when the design phase is completed, the metadata is sent to the CTR database developer. All coding and development occurs at the build stage, which is undertaken in a restricted access development environment. When work is released for testing, it's deployed via a test environment, and testing is recorded on a test script template. Formal user acceptance testing is completed when all data fields, export, so checking the data entered matches what comes out, audit, so this is checking the data changes export and record a reason for change, and security tests, so user access, is set to pass. Throughout the life cycle of a study, the metadata may need to be amended, and this could be due to a metadata error or a protocol amendment, and then we would move back to the design phase. So once the build is past testing, it can be put to live. The CTR release to live procedure is intended to ensure that all documentation is in place and the software package to be released is the same as the software that was tested and accepted by users. Checks are also completed to ensure data can be archived in line with the retention time specified in the trial protocol. So once live, user access can be granted. As per example 1, the delegation log shows who can enter data at site. Investigators are given access to the database once the trial is open to recruitment, and data is returned prior to final database lock. All users are trained in the system's data flow, entry, form triggers, validations, data, and data query resolution. And this is done via site initiation visits and hard copy guides and/or training videos, and extracts are an example too. CTR systems are hosted on the Cardiff University's Resilient Network. If databases do become unavailable for whatever reason, manual paper-based forms are used, and this data is entered onto the system once available, and backup processes are in training guides, and this is shown in example 3. The CTR uses both bespoke software developed in-house, such as TMS2 for complex randomization, or commercial off-the-shelf software. So external systems include Macro and more recently REDCap, for which we were part of the GW4 collaboration to validate REDCap and develop a data entry user guide. All software goes through a CTR software validation risk assessment, which considers users, maintenance, license costs, needs, and testing requirements to aid selection. Notwithstanding, we do still use Excel for some data collection, such as screening data. So the first database case study is Tiptoe, which will be summarised in a very short video by CTR data manager Ceri Nash. So Tiptoe is a living well support package for people 65 or over with knee and/or hip joint pain and long-term conditions. And the trial implemented a user-centred design process to support 3 levels of access in a decentralised trial. So hopefully now you'll hear from Sarah.
Tiptoe uses 2 secure databases, REDCap as the data repository storing all the collected data, and Assistant database designed by an independent, Baseline Software Limited, which is the user interface. Assistant does not store any information, it is only a view. It pulls the information through from REDCap in the background using an API, an Application Programming Interface, which is the connection between the two databases. REDCap securely holds all the data. Participants register their interest by completing the online registration form to check they are eligible for the study. When the form is submitted, REDCap creates an individual record, an identifier, so the next sequential number for the participant. The assistant system automatically sends an email to the participant to let them know that they are eligible or not eligible. If eligible, assistant sends a link to the participant for them to create their own password to use to log into the system, provide their consent, and complete questionnaires. The participants start their journey through the Tiptoe website and as an online self-referral, other routes being in person, in clinic, or over the telephone. They can read the study information, register their interest, and contact us through the website. They use this platform to view, read, consent, and complete questionnaires. The study team and sites use the Assistant database system. They log into Assistant and are able to view and manage participants, and sites PIs are able to allocate participants to their practitioners. Practitioners are able to view participants allocated to them and independently manage and book intervention sessions. Assistant has been further adapted for Tiptoe to include a reporting tool whereby graphs and charts are created to show recruitment overall, per site, and per month. Tiptoe uses...
Okay. So the second case study is SunPad. So this study is conducting a comprehensive field test of safe, reusable menstrual health products with material developed by the Cardiff University chemistry team in Nepal, where access to clean water is limited. 50 experienced reusable pad users will be asked for feedback on their experiences and thoughts after using the product, and the pads will be analysed in the laboratory to see whether replication of laboratory findings was possible. So SunPad sites have limited or no Wi-Fi, so our normal database solutions wouldn't work. REDCap has an app for tablets and iPads, which allows for offline data collection. However, where data is collected without Wi-Fi, and indeed for any REDCap app software version updates, iPads need to be synced with the REDCap database via Wi-Fi. So in remote areas, this could lead to delays of several days. Whilst the data collection isn't particularly complex, the additional steps researchers need to follow on the iPads is. So this photo just shows members of the CTR team training members of the team in Nepal as we provide a high level of ongoing support. So just to summarise, CTR has robust systems to ensure databases are designed and built in accordance with ethical and regulatory requirements to answer the study question. Validations ensure data is clean and complete for export and analysis. CTR database designs and builds are collaborative, so internally multiple CTR departments work together, and externally working closely with CIs, PPI and other CTUs such as GW4, and the REDCap data entry publication output link is on this slide. As for the two case studies demonstrated, CTR databases support adaptive trial designs and focus on delivering reliable, clean and complete data for analysis. So I'm just going to stop sharing and hand over to Debbie.
Thanks, Ceri. Bear with me. Can you all see that?
Yes, thank you.
Perfect, thanks very much. So my name's Debbie Harris. I'm the Senior Data Manager for the Infection, Inflammation and Immunity Division, otherwise called the I3 division within the CTR. So how does good practice in relation to data management translate into our trials? As well as it being good practice to adapt data collection to change in legislation, it's also good practice to evolve with changing times and circumstances. So in my section of the presentation, I'm going to cover some examples of how we have achieved this within the CTR. This will cover how our data collection has evolved over time, how much data to collect, how we gather evidence for changes to data collection processes and methods, and also looking at not just trial data. So when I first started my role as a data manager in the CTR 11 years ago, electronic data collection was in its infancy. Trial data was collected on paper forms, which were then sent to the CTR for entry onto Access databases or Excel spreadsheets. However, now data for all trials is entered onto study-specific electronic databases, and as we have heard, we use a variety of these within the centre. In the early days of electronic databases, data was still collected on paper and sent to the centre for entry onto the database by the data managers. This has now progressed, and study staff at sites are able to access and enter the data onto the databases themselves, reducing the need for paper forms. These electronic database data collection methods are also continually evolving with the times and needs of the study. For example, during COVID, social distancing meant face-to-face follow-ups could not take place. So we had to find a different way of completing participant follow-up so that data was not lost. Our database developers updated our databases so that participant-facing forms could be completed via email links sent to those participants who had provided an email address and were happy to be contacted in this way. So this is an example of how this looks in our databases. If an email address has been provided by either the participant or, in the case of this study, an alternative contact, the forms can be sent by the click of a button. The system will then show if an email has been sent and when. The participant will then receive an email, and within the email is a link to be clicked on. And once you click this link, this then opens the questionnaire for completion by the participant directly into the database. Participants do not see any other data from themselves or other participants, and once the forms have been submitted, the link is disabled. And this has now become a standard feature of our in-house databases for studies that would like to use this functionality. We are now evolving further in using apps for participant-reported data collection. One of our new studies, Balloon, will be using an ePro app for participant-reported outcome measures. We are working with a company called Your Research, who create the app to develop the forms and data that we will be collecting. Parents will be able to download the app free from the Apple App Store or Google Play Store and complete the data entry in real time. This is fully customizable to the study needs, and for Balloon, we will be using this to track adherence to the study medication, healthcare usage, amongst other things. So as data managers, one of the things we have to consider is how much data we are collecting. It's not ethical to collect too much data, and we should only be collecting enough data to answer the research questions as listed in the trial protocol. However, there is always the temptation to collect too much. In 2020, this paper was published, and the aim of this was to categorize the types of data collected and to look at what proportion of collected data each category represents. The results of this study show that primary outcome data accounted for a much smaller proportion than secondary outcome data, but the largest amount of data collected was non-outcome data. We've taken this paper as an example and set up a data justification group within our data managers to see if we can use data categorization to reduce the amount of data we collect in our trials. And as has already been mentioned, every variable in our trials metadata now needs to be justified using one of our 8 categories shown here, with the aim that if a variable does not fit into one of these categories, it should be discussed among the trial team if it is needed at all. This hopefully is leading to less data collection, which ultimately has benefits for all concerned. There will be less burden on site staff and participants to collect and enter their data, and less burden on the data managers by reducing the amount of data that needs to be cleaned and queried. I just want to mention the importance of public and patient involvement, otherwise known as PPI, in data collection. The definition of PPI is research carried out with or by members of the public rather than to, about or for them. We have PPI representatives on our trial management groups and who we work in collaboration with in all aspects of the trial. This includes the planning, designing, managing, conducting, dissemination and translation of the research. PPI involvement is extremely valuable in providing a different perspective to data collection in our studies and can help prevent the wrong questions being asked by providing focus on the areas that patients consider important. This includes advising on what data we are collecting and what methodologies that participants are more likely to engage with to improve our data collection and quality of the data. And just as an example here, a previous study, Domino HD, received an award for its PPI involvement. And if anybody wants more information on PPI involvement in trials, please take a look at our website. So in order for any changes in data collection processes to be implemented, there needs to be evidence that the change will be of benefit. To gather this evidence, trials can make use of SWOTs, which are also called studies within a trial. A SWOT is a type of a study in which an intervention to improve the conduct of a trial is tested in the context of an ongoing trial. These can be generalizable to other studies or be of specific benefit to the host study itself. And SWOTs are becoming more common in our trials within the centre. And we have incorporated SWOTs in some of our studies to look at different data collection methods and also to evaluate methodologies that can increase follow-up data collection and participant retention in our studies. I'm just going to mention 2 examples of SWOTs that are being carried out in the CTR. So Rhys Williams-Thomas created a SWOT looking to see if different designs of patient diaries could impact the data response and quality. So Rhys used 2 studies for his SWOT, which were PACE HD and SANITA. And he developed 2 diaries, one using the standard electronic interface shown here on the left, and the second with the new design on the right. Participants are randomized to receive either the old or the new diary, and at the end of the study, participants will complete a user experience questionnaire, in addition to a comparison between the numbers of diaries completed and their completeness between the 2 groups. Secondly, placement is a study currently open and recruiting. We have a SWOT embedded in the study to see if video information improves participant follow-up rates. So a few years ago, we undertook the placement feasibility study, where follow-up rates were low at less than 40%. So in order to try and improve on the follow-up rates in the full trial, participants, if willing to take part in the follow-up SWOT, will be randomized to receive either a follow-up information leaflet alone, shown here on the left, or shown a short video about what the follow-up will entail, when it will be, how long it will take. And the hypothesis is that those shown the video will engage better with the follow-up than those given the information sheet alone. For anybody interested in finding out more about SWOTs, the Queen's University Belfast in Northern Ireland hosts a SWOT repository, and SWOTs can be viewed in the repository so that anyone can see if an idea for a SWOT has already been developed, which can help to avoid unnecessary duplication of work. And the repository can also provide SWOT ideas for other trials to either adopt or adapt themselves. And another good source of information on SWOTs is the Trial Forge SWOT Centre, University of York, which has been working to develop resources on the design and delivery of SWOTs. So finally, I just want to mention that not all data we collect or need to manage is trial data, but is equally as important. So in another example of developing new processes depending on the needs of the trial, we are currently working with our database developers to build a randomization system that is linked to an IMP management system. Previously, we have used external sources for this purpose, which can be costly. So this is currently being developed for 2 studies, SAMADI and Balloon, and the system will allow participants to be randomized to different arms of the study to either active or placebo medications based on the trial pack allocation. And it will track the supply of medication from our manufacturers to sites, onto participants and back again if necessary. It will also track the stock of IMP at sites and can issue resupply of stock when necessary. So in summary, we are constantly developing new, more advanced data collection methodologies, and this will hopefully lead to improved participant and site experience in trials research. This will lead to improved participant and site engagement with the study, which ultimately will lead to improved data completeness, accuracy, and thus data quality. As without reliable data, we cannot provide conclusions to our trials. Thank you for listening. So I'm now going to hand over to Kim, who's going to talk about routine data collection.
Thanks, Debbie. Right, I'll just share my screen. So doing the standard check, everyone can see my screen okay?
Yes,
yeah.
thank you.
Yeah.
Fabulous. Hello everyone, my name is Kim Mannery. I am a routine data study manager and I also work at the Centre for Trials Research. I was also a data manager previously within the centre. And today I'll be talking to you about using routine data in trials and the importance of good governance as well. Sorry. Okay, so most of you may know already, but routine data is information that is captured when people use different public services. So this could be the NHS, different social services, schools, college, university, prison, courts, transport. There's, of course, lots of different public services. So this is where data is captured there. And researchers can use this type of data in different studies, such as clinical, interventional, observational studies and feasibility studies as well. But it's important to note that routine data is collected for service use and then has a secondary, usually a secondary usage for researchers. Okay, so routine data and its importance then, and its benefit. So as mentioned, it's an existing resource. It's already there. As soon as someone uses a service, that data is captured. Therefore, it's very rich in information and has a lot of variables that could be really helpful for research. And they often cover a large population and also a diverse population that sometimes trials find it tricky to recruit. So we do have to be mindful as well that this resource that we're discussing, routine data, is based on populations that use services. So people who don't use services will not be within these data sets. So it can inform study design and it can help with, routine data can help with calculating sample sizes. It can support smaller feasibility studies where resources are limited, especially in terms of data collection. And it can also help to understand your research population further, especially at the pre-planning stage, so before you've even had any approvals. So objectivity with routine data. So because it is collected as part of a service, it can provide an objective assessment of an outcome or a characteristic. Routine data can also be used to validate with self-reported data in trials. Because sometimes trial participants may not want to fully disclose interactions with services or certain type of events, and bias can come into play sometimes with self-reported data. So this routine data can be objective. And it can support patient or participant recruitment. And this is because it's using routine data can reduce the burden, as discussed in this presentation, for general trial staff, participants, just to reduce that contact time and the amount of data that's being collected within the trial. It can also help with the emotional burden as well when discussing sensitive topics. And with patient recruitment, it can support identifying numbers of certain populations if that's within your research question, where you're looking at specific cohorts and not the general population as such. So it can really inform an inclusive trial design as well. And with site selection, that's being used more recently, actually, where routine data can support site feasibility checks. So that's another good way of using routine data itself. Data completeness, so linking routine data with other data sources, like data collected in trials or other routine data sets that may be in a different area, so not health, it could be education, and that might enhance your at the end your trial data set that is being analysed. And also using routine data can also help with looking at your trial outcomes and can be used for trial outcomes. So it can also help with loss to follow-up if participants, you are no longer able to contact them. As discussed, because it helps with loss to follow-up, it does generally help with long-term follow-up because it reduces the burden on participants that are involved in your trial and reduces potential visits to sites, and is often used in longitudinal studies as well, routine data. So generalizability, it can be used to look at the general population as a whole. And as discussed, this is people who use services. And it can often, because with trials, the challenges with recruitment, it can also, again, bring in more diverse data in your trial data. Okay, so as discussed throughout this webinar, it is imperative to understand your data and the data protection rights for participants and for everyone. So data protection and privacy should be considered even before the processing of any personal data, as Nigel has mentioned. And this is whether it's from an existing routinely collected data set, or if it's a self-reported trial, comes from self-reported trial data. So UK regulations are there to set standards on how to safeguard the public and the quality of the data being collected, also in turn enhancing the quality of the research. So if these particular regulations, such as the Data Protection Act and GDPR, are not followed, it can cause issues with your trial and sometimes be stopped if it's been audited. So with the Data Protection Act and GDPR, as already discussed, there is now greater focus on transparency and accountability. And this increases our responsibility as an organisation and individuals who are working with personal data. So the different documentation mentioned in my slide is, of course, really important for data standards, and it ensures consistency and creates linkable data that can then be linked with other data sources, so you can track and trace, which is similar to what Helen discussed with the storytelling of your data throughout your trial. So the number one thing is that it's essential to understand the different categories of data. So special category data that I've mentioned in brackets is personal data, which requires much more protection. So this data should be processed in limited circumstance, and there should be clear justification for why it's used. And these documents that I've mentioned, data protection, impact assessment and data sharing agreements, data management plans and cleaning plans, are all really important when you're looking at how you'll manage your data within your trial, especially data flows where you're working out where identifiable information is being transferred and whether you can mitigate against potential risks of identifying participants or people within these routine data sets. Next slide. So the common law of confidentiality. So this is basically a law within the NHS laws, which is called Section 251. And basically that means if you want to use confidential patient information, you must go through a public approval panel first to basically disclose exactly what you're going to be doing with this personal data, and mainly to ensure that you're safeguarding the public and safeguarding them from risk of re-identification. everything I've discussed is very, it ranges from a level of being fully anonymized, the data sets, and then going downwards, looking at more disaggregated data, so more personal data that special category data would be within that. So section 251, when you're going to a public panel, you could either go through the confidentiality advisory group CAG, which is Wales and England specific, or if it's a slight change of law in Scotland, but you still go through a public panel where they approve your data management, basically. So that would be the public benefit and privacy panel. And just to mention, as GCP has been mentioned as well, GCP has been updated and they've included the use of electronic health records. So it's always good to have a look at that. And I believe it's mentioned in the hot topics section. So thank you very much, everyone. I just want to say that routine data is, of course, a rich, powerful tool, data collection tool. And when used safely and ethically, it's extremely useful and can improve trials for participants, trialists, and the public. And it enhances our understanding of the society as a whole and where change is needed. And just a quick add, basically, the TopCat project that we're working on at the moment, we're developing learning resources for people working on trials for using health systems data particularly. So please do check that out. Thank you.
Thank you.
Thanks, Kim. So just a few concluding thoughts from myself. So hopefully today we've shown you the importance of data management and making sure that we're getting the right data, the fundamentals of what we do and why we do it that way, the importance of our databases and the different ways that we've set them up, how we try to innovate our data collection, so not one trial is the same, so making sure that we're always doing the best for that specific trial, and also the growing use of routine data in trials and the benefits of this. So I think Richard has just put a feedback link in the chat, and I think he'll be sending it out by email as well. So it'd be great to hear your feedback on this webinar. I am conscious that it's just gone 12 o'clock, but I think we can take maybe one or two questions before we dial out if people have any. No, that's fine. I think if you do have any questions, I think you've got the Centre for Trials Research's email address, so please contact us there and one of the presenters will get back to you. So thank you very much, everyone, for attending today.
Thank you.
Thank you. Thank you.